phase is called interphase, in which a nuclear envelope surrounds uncondensed chromosomes. At this point, they're duplicating and growing and duplicating themselves. There's two centrosomes in the cell, and if it's an animal cell, they have centrioles in the middle as well. Then we move to prophase, in which the chromatin condenses. First, the nucleoli disappears, and the chromatin is replaced by fully condensed sister chromatids that are attached to each other. The mitotic spindle begins to form from the centrosomes, and asters, which provide structural support, as well as microtubule arms extend. Next is prometaphase, in which the nuclear envelope fragments entirely, and the microtubules extend, attaching to the sister chromatids at the kinetochores, which are structures in the middle of the chromatids. Some of the microtubules are called non-kinetochore microtubules and do not actually attach to chromatids. Then we move to metaphase, which is the longest stage of mitosis. The centrosomes are now fully on opposite ends of the cell, and the chromatids line up at a divisible line called the metaphase plate. They're still held together at the kinetochores by the spindle arms. Then comes anaphase, which is the shortest stage of mitosis, and the microtubules shorten, which pulls apart and separates the sister chromatids, forming fully fledged chromosomes. The cell begins to elongate, and as two new cells begin to form, you can see that each of them has an equal number of chromosomes. Then is telophase, in which the daughter nuclei form again, as well as the nuclear envelopes, and the genetic information is genetically identical. And finally is cytokinesis, which is the division of the cytoplasm. In plant cells, a cell plate would form, which would eventually become a cell wall. But this cell is an animal cell, so a cleavage furrow forms, pulling apart the cell entirely, creating two new genetically identical cells. This is mitosis.